Good day, everyone, and thank you, OIS, for allowing us to be here today to provide an overview of our Archer Phase 2 study in geographic atrophy. We'll be making forward-looking statements as part of this talk. So at Annexon, we're focused on blocking classical complement activity upstream on diseased tissue. And it's a really important distinction from prior complement approaches because we're blocking on tissue where disease emanates from to provide functional benefit. We've done so preclinically in the GA space where we show protection against photoreceptor cell loss and function. And the Archer Phase 2 study is the first clinical program evidencing that mechanism of action where we showed significant dose and time-dependent visual preservation on BCVA 15-letter loss. There are multiple lines of evidence in support of this visual preservation. There's the on-treatment a portion of the analysis. We also did a fellow eye analysis as well as an off-treatment analysis, and we'll show you all of that. Suffice to say, based on that data, we were granted the first and only program to receive EMA Prime designation, and we're now actively preparing a global Phase 3 program to confirm the findings. Now, importantly, photoreceptor cell loss, synapse loss, and function occurs prior to RPE loss in geographic atrophy. As you can see on a cross-sectional of the retina of a GA patient here on the far right, where the RPEs are designated in fluorescent green. When you lose your RPEs, that is where you have lesion growth. The green arrow at the bottom represents the growth that you would expect over the course of a year from the loss of RPEs. And the currently available therapies in GA impact roughly 20% of that on an annual basis. Importantly, prior to the loss of RPEs, you have loss of photoreceptor cells and synapses. And so when you look at the far left and move right from left to right, in blue are your photoreceptor cells, and in red are the photoreceptor synapses. And as you can see over the course of the retina, those become increasingly degenerated prior to the RPE loss, such that when you do see loss of RPEs, above there, you have virtually no synapses left or no photoreceptor cells. That represents function. That is where your vision is housed, and so that mechanism occurs prior to RPE loss, and that is the focus of Annexon in targeting C1q and the classical pathway. This slide here just depicts our mechanism of action and what the company was founded on, which is, again, blocking upstream classical complement activity on diseased tissue. Here you can see C1q is localized and anchored on functioning or photoreceptor synapses, anchoring the classical pathway and driving this inflammatory disease process down through C3, C5, C9, et cetera. Only by blocking C1q can you block this entire disease process. Indeed, blocking C3 and C5 allows the upstream complement activity to remain in place, resulting in inflammation and tissue damage. We've shown this preclinically, which was quite important for us to receive Prime designation in Europe. Here on the left, we see C1q in green localized on synapses in the red in a post-white light damage model after day 3. On the right, in published literature, by blocking C1q with our intravitreal administered Fab ANX007, we were able to protect photoreceptors as well as their function in this model. So we've taken ANX007 further into the clinic, which we're quite encouraged by. This is a small Fab, 50 kD. It has low viscosity, non-pegulated, and we demonstrated in prior patient studies the ability to inhibit with full target engagement in the aqueous humor. Turning to Archer, the Phase 2 itself, it is a Phase 2, 270-patient randomized double-masked study. It included both foveal and non-foveal lesions. We allowed patients with CNV in the fellow eye, and it was randomized 1:1:1 between sham every month and every other month with 5 mg dosing. It's 12 months treatment and 6 months off treatment. Starting with the functional endpoint of vision, BCVA 15-letter loss, this is a widely accepted assessment of visual acuity. In fact, it is the gold standard for visual acuity in the ophthalmic space. It's been used for multiple approvals in this space, some examples here on this slide. And importantly, it represents 50% reduction in vision capabilities over a 1-year period of time. In our study, we assessed BCVA 15 on 2 consecutive doses. We're at month 12 to ensure veracity and to minimize any variability in this measurement. Looking at the data, we were really quite pleased to see that ANX007 demonstrated a highly statistically significant protection against vision loss in the every month group, and we showed a very nice trend in the every other month group versus sham. We also did a time to treatment analysis as well, where we showed a 72% reduction in loss of 15-letter vision in a 12-month period of time in the every month group, and roughly 50% in the every other month group. Importantly, the treatment improved protection against vision loss over time. So as you look at the second 6 months in the study, we saw an increasing benefit of the drug with longer treatment, which we think bodes well for continued durable effects with regard to this approach. Now, we also did a fellow eye analysis. This is in the patients in the study who had bilateral GA, that is geographic atrophy in both eyes. In the patients who were treated, you can see in the sham group, the patients with study eye and fellow eye, virtually no reduction in preservation of vision loss. However, in the every month group, what we saw was a 74% reduction in those patients who were treated in the study eye versus in their fellow eye, and nearly a 50% reduction in the every other month group. Again, consistent with the overall population, but this being a subpopulation of those with bilateral GA. We were also encouraged to see that this impact was across both non-foveal and foveal patients, speaking to the veracity of blocking the loss of photoreceptor synapses and preserving photoreceptor cells themselves. And then finally, looking at an off-treatment analysis, we're really encouraged to see that the protection that we demonstrated over the 12 months period of time really set a new baseline for the patients. And once you stop treatment, that treatment effect began to wane, and the patient's progression really mimicked the sham arms. And so encouraged by that and think that there's really a strong, durable biological rationale for what we're seeing with this effect. We, of course, also looked at fundus fluorescence and lesion growth, the primary endpoint in the study, and there we did not see statistical significance, but again, bear in mind that is an event that we'd expect to occur after the loss of photoreceptor cells and synapses. And with longer treatment, we expect that we will have a greater impact on lesion growth. In fact, when you look at the second 6 months of the study, we saw exactly that. So with longer treatment, we expect that we will do quite well there. From a safety profile, ANX007 was well tolerated in the study. There was no observed differences in the CNV between the sham and the treatment arms. There were 3 cases of endophthalmitis. You can see a case of retinovascular occlusion, no cases of retinovasculitis, no cases of ischemic optic neuropathy. And so overall, we're quite encouraged by this neuroprotective approach. This is our 3rd demonstration of protecting synapses to provide neuronal health and functional outcomes. We did it previously in the brain, same mechanism, just different compartments in Huntington's disease and ALS. And so we're encouraged to see this demonstration in the eye. And as I said, we're looking forward to initiating a phase 3 global program next year. Thank you.